Okay, so actually we were discussing microbiology and microorganisms. We are going to discuss in these particular classes. So we were discussing prions. Prions are protein infectious particles. So the term is revealing PR for prion and ION for infection. Okay, so when these infectious particles are proteins, so this was a new class identified which can also cause some diseases, and these diseases are varying from organism to organism. Okay, so actually the reason being the same, most of the prion diseases are happening because of the conformational change in protein. So generally, it is seen that in most of the cases, if the alpha form of protein is getting converted into the beta plated sheet, so it will lead to some abnormal condition, and these diseases are called as prion diseases. These prion diseases are named according to the organism in which they are happening. For example, scrappy was first reported in sheep. So what was happening, what was the observation was that if the sheep is having this disease, so the sheep will start rubbing his body against the wall or fence, which ultimately causing the loss of wool. Okay, so first time when the people uh, who actually have these sheeps, they noted or observed that there is a loss of wool because these sheeps will start rubbing their body against the fence or wall. Therefore, ultimately, their wool will be lost. Okay, so this type of disease then observed in different other organisms, in fact in humans also. And in humans, it is known as Creutzfeldt-Jakob disease or simply Kuru. And one more disease is actually mad cow disease. If a human some human eat the brain of infected cow, then the human can have mad cow disease. So different reasons and different type of organisms, based upon the organism in which the disease is happening and depending upon which is the donor, the disease may have different names. Okay, but the major thing which is common to all the prion diseases is the infectious particle, that is protein here. Fine. So before starting microbiology, because microbiology is so interesting to discuss things, two reasons why I'm teaching microbiology before immunology. I should start immunology first, but okay, so the two reasons I will discuss why I'm starting microbiology first. So the microorganisms were, the first reason is because microbes were evolved first over Earth about 3.5 billion years ago. So actually our Earth was formed around 4.5 billion years ago. After about 1 billion years, so different single cellular organisms that are called microorganisms or microbes start appearing, and actually these are microbes. Second thing is because in immunology we will discuss different defense mechanisms of our body, different types of cells. For example, B, T, and NK cells, which are the major cells, but in fact macrophages and neutrophils are also important in immunological responses. Okay, so generally we call them neutrophils are called chote haldar and macrophages are called bade haldar. So these different type of cells are also important to eliminate these microorganisms. So first of all, we will discuss all the pathogens, all the microorganisms which can cause the disease, and then only we will start our discussion of immunology. Okay. And before starting the topic, actually there is an interesting story. Okay, so about prions only. So if you have heard about or not, I do not know, but actually this is an interesting story. So let's start. So, So actually, when the humans start eating humans, this is a ritual in some of the tribes actually, and this is called cannibalism. Okay, there are different uh, traditions in different communities and tribes, in fact, and cannibalism is one of them. So when societies throughout history have practiced this type of activity when humans ate humans. So the reason for this ranges from the belief that eating another person allows their powers to be absorbed. So some people or the some societies believe in this that if they eat another human, they can absorb their powers, their okay, so their brains will come into them and they will have more power, in fact, to a way of overcoming the death by allowing the individual that is consumed to become immortal. Or in some societies, the humans were consumed because they think like they will become immortal if they consume the other human beings. To this day, the Aghori, these Aghoris, so they even today they practice this. People in India, they still eat their dead because they believe that eating the dead humans can help them overcome the fear of death. So there are different type of beliefs, and there are different diseases which have been evolved in the society. Okay, so there are others also, other rituals, for example, human sacrifice and so on. You will get Sati Pratha and many different. Okay, you will find this over internet. But actually, our discussion is about cannibalism because this type of tradition have resulted uh, in the disease that is called Creutzfeldt-Jakob disease or CRU disease in humans. Okay, so there is a query. I do not know what you want to know, Neharika. Okay, so Neharika ma'am, I will tell you in Hindi. You please call me after the class and I will explain in Hindi. Okay, because there are different type of students in this particular class, so I need to teach in this language only so that all are able to understand. Please cooperate. So now,
नहीं
okay,
हेलो 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 कृष्णा
yes,
हेलो थोड़ा सा यूज कर लो पूरी नहीं बोल रही है थोड़ी बहुत
sure. The major target of this class is to understand. Okay, okay, so if you are not able to understand, the class will be waste. So I will try. Okay. So ये जो प्रथा थी, ये जो traditions थे, means for example cannibalism, इसकी वजह से ये diseases, for example prion diseases in humans, that is also known as Creutzfeldt-Jakob disease or CRU disease, evolved हुई. Okay, so here, okay, तो यहाँ पे एक और note है. So there was a tribe, and most importantly, as one of the medical researcher described, if the body was buried, it was eaten by worms. So देखो, अगर सिंपल सी बात है, अगर किसी की बॉडी को अपन दफन कर देंगे तो क्या होगा, या तो उसे वॉर्म खा के खत्म कर देंगे, और इट वाज प्लेस्ड ऑन द प्लेटफॉर्म, इट वाज ईटन बाय मैगोट्स. और वहां पे जो है छोटे छोटे माइक्रो ऑर्गेनिज्म और मैगोट जनरेट हो जाएंगे, दैट दे विल अल्टीमेटली डिग्रेड दैट पार्टिकुलर डेड बॉडी. Four believed it was much better that the body was eaten by people who loved the disease than by the worms or insects. तो कुछ ट्राइब्स ऐसे हुए जिन्होंने इस बात पे ज्यादा जोर दिया कि क्यों ना उस बॉडी को, मान लो कोई हमारा बहुत ही ज्यादा लव्ड वन मीन्स कोई परिवार का कोई सदस्य है या कोई क्लोज रिलेटिव जिसकी डेथ हो गई है, उसको बजाय वॉर्म्स और इंसेक्ट को खिलाने के क्यों ना खुद से ही खा के खत्म कर दिया जाए। ठीक है। सो इस ट्राइब में एक्चुअली क्या होता था, वुमेन्स रिमूव द ब्रेन, सो दिस वर्क वाज गिवन टू द वुमेन। नाउ देयर वाज आल्सो डिफरेंट बिलीव्स आफ्टर दिस, बिकॉज इट वाज बिलीव्ड दैट कि जो महिलाएं होती हैं, ठीक है, क्योंकि they give birth, so it is their moral responsibility that they should also eliminate that particular organism or individual after death. So this was one belief, and the second one was कि जो जो ये जो शक्तियां होती हैं, ऊपरी ताकतें होती हैं, वो नारियों को अफेक्ट नहीं करती. So women's these women removed the brain, mixed it with the fawns, cooked it in the tubes of bamboo, and they fire roasted and ate everything except the gallbladder. So अब देखो डिफरेंट डिफरेंट बिलीव्स ही हैं जिनके बेसिस पे अलग अलग ट्राइब्स जो हैं उन्होंने रिचुअल्स बनाए हुए हैं. तो वो लोग क्या कहते थे कि गाल ब्लैडर को छोड़ के वो पूरी बॉडी को कंज्यूम कर सकती थी. सो सबसे पहले ब्रेन को खाया जाता था. ठीक है. इट वाज प्राइमरीली एडल्ट वुमेन हु डिड सो, एंड दिस वाज एक्सप्लेन्ड बाय दिस पार्टिकुलर मेडिकल रिसर्चर. Okay, because their bodies were thought to be capable of housing and tamping the dangerous spirits that would accompany a dead body. क्योंकि ऐसा माना जाता था कि जो वुमेन होती हैं, जो ओके, तो वो उन बुरी शक्तियों को, उन बुरी आत्माओं को अपनी बॉडी में हाउस कर सकती हैं और उन्हें स्थान दे सकती हैं. Okay, so the woman took the role of consuming the dead body and giving it a safe place inside their own body. Okay, so for a period of time during this dangerous period of mortuary ceremonies, uh, this kind of ritual was a ceremony in which they consume the dead ones of their family or in their relatives. But women would occasionally pass pieces of feast to the children. But actually क्या होता था कि जो लोग means जो man होते थे, they went to the jungle because it was a tribe, so in search of food. So generally वो लोग शिकार करते थे और generally शाम को ही आते थे या जंगलों में ही रहते थे, ऐसे बोल सकते हैं. लेकिन क्या होता था कि जो अगर किसी की डेथ हो जाती थी तो घर में जो महिलाएं होती थी और छोटे बच्चे होते थे, तो कई बार क्या होता था, जैसे महिलाएं उसको रोस्ट करती थी, उसको पीसेस में कट करके कंज्यूम करती थी, तो वो छोटे छोटे पीसेस कई बार तो बच्चे होते थे मांगते थे तो उन्हें पास कर देती थी. ठीक है, एक छोटी फिंगर दे दी जैसे. ठीक है. सो दे पास पीसेस ऑफ फीस्ट टू द चिल्ड्रन. They ate what their mothers gave them because they were children, they know nothing, and generally the small children ate the pieces which were passed to them. Okay, until the boys hit a certain age and went off to live with the man in the jungle, then they were told not to touch that stuff. उसके बाद में क्या होता था कि जब वो बड़े हो जाते थे तो फिर वही, क्योंकि दिस वाज अ वर्क ऑफ वुमेन ओनली, सो जो महिलाएं होती थी वही उस मरे हुए इंसान को कंज्यूम करती थी. तो बाद में उन्हें बोला जाता था कि अब वो उसको टच नहीं करेंगे एक बार वो जैसे घर से बाहर चले जाते थे. सो दीज वर रिचुअल्स एक्चुअली. Now what happened in that particular society or in that particular tribe after some years, because these prion diseases are having some incubation period of around uh, 10 years or so. So the children were the death of these children who consumed those pieces actually started due to prion diseases. So finally, after urging for research, researchers like, so these type of medical researchers went to that particular tribe and they tried to understand the molecular nature of those particular pathogens. So biologists from came around the world with this idea that the strange disease stemmed from eating the dead people. Uh, okay, so this was understood that देखो कि कुछ ना कुछ तो हो रहा है अगर ये डिजीज फैल रही है और जो छोटे बच्चे हैं जब तक वो अपनी एडल्ट एज में पहुंच रहे हैं यानी 15-20 साल के हो रहे हैं उससे पहले उनकी डेथ हो जा रही है तो एक्चुअली क्या हुआ कि उस पार्टिकुलर कम्युनिटी में उस पार्टिकुलर ट्राइब में केवल बूढ़े लोग ही रह गए जितने भी नई जनरेशन थी उन सबकी डेथ हो गई तो उन लोगों को एक चीज तो समझ में आ गई ये जो रिसर्चर्स थे कि एक्चुअली ये लोग जो डेड पीपल्स को कंज्यूम कर रहे हैं उसकी वजह से इनकी डेथ हो रही है लेकिन till now nobody is aware of the fact that how this is happening. So the case was closer after a group that US National Institute of Health NIH injected infected human brain into the chimpanzee and was the symptoms of croup developed in the animals months later. So because this is having large incubation time, so it took months to activate the prion particle and the disease symptoms appeared first. The group which won the Nobel Prize for the findings, okay, so dubbed it a slow virus, but actually it was not the virus. Even not a bacterium, not a fungus or a parasite. It was entirely a new infectious agent, one that has no genetic material because it was having either not the DNA, ना तो उसके पास DNA था, ना उसके पास अपना RNA था. Only protein, and this protein can cause the disease. In fact, this protein can propagate from, so it will continue to infect the, okay, so if someone consume the diseased animal or part of that particular organism, he or she will be diseased. और एक चीज और could survive being boiled and was not even alive. So पहली बात तो क्योंकि ये केवल प्रोटीन का बना हुआ एक infectious particle था, so it was not alive. Second thing is if you will boil or will roast this particular flesh or the part of organism in which this particular protein is present, so it will survive the boiling even. Okay. And in the deer or an elk, actually this disease is the same disease because this is also prion disease. This is known as chronic wasting disease. Okay, so there are different names for the same disease. Ultimately, what happened, how it is a lethal one, because the protein conformation will change from alpha to beta pleated, alpha helical to beta pleated sheets, and it will lead to the compelling the normal proteins on the surface of nerve cells because there are so many proteins which are present in the nerve cell plasma membrane in the brain and therefore called proteinaceous infectious particle would eventually misfold enough proteins to kill the pockets of the nerve cells in the brain and leaving the cerebellum riddled with holes like a sponge. So ultimately what happened, so your brain or this particular portion of the brain called cerebellum will be converted into a sponge-like structure. There were pores were created and the normal functioning will be interrupted. Okay, so again we are moving to okay. Here. Okay, so first thing is the microorganisms or microbes are causative agents of many different diseases. For example, plague, which killed a major human population around 1347, also known as Black Death. But some of them are important also because they will help us to digest the food and produce some of the vitamins which are very important micronutrients in our bodies. For example, vitamin B and K. This K is important in clotting actually. So the blood clot. And various vitamin B, for example, vitamin B6 and other different type of vitamin B are important because they are important portions of coenzymes. We have discussed already this in our enzyme class. Okay. And the other microorganisms which are present in the gut of herbivorous animals, helping the herbivores to digest the grass and leaves. Because cellulose digesting enzymes is not present in animal kingdom, only with the help of these bacteria or these different type of microorganisms, they are able to classify, able to degrade the cellulose. Also, the termites, which degrade the wood, also have these bacteria in their gut so that they are able to get their energy from the cellulose of wood. Then there are different, if we try to classify different type of microorganisms, they can be classified into two major groups. One is called cellular, one is called acellular. So actually we had discussed prions. Prions are coming under acellular because they do not have any genetic material or, so they are completely different. Instead, they are different classification. It means viruses, viroids, virusoids are also acellular entities. Okay, some cellular microorganisms are bacteria, that is today actually called eubacteria, archaea, single cell eukaryotes that are called protists or protista, and fungi. Fungi, you can classify them either into unicellular yeast or multicellular mold. Okay, so they can be classified like yeast, mold, protists like algae, protozoa, protozoan parasites. We have discussed in case of African sleeping sickness, slime molds, and so on. Bacteria, for example, E. coli, Staphylococcus aureus. Okay, so many different bacterial bacteria we will discuss. Archaea, these are completely different because archaea can survive under harsh environmental conditions. For example, if there is very high temperature, very high pressure, so where most of the cellular life cannot survive, archaea is able to survive those harsh conditions of environment. For example, methanogens. Now the question is, because we are having different type of microorganisms, as we have discussed in our 11th and 12th class, so of Whitaker's Five Kingdom classification system, then this Five Kingdom classification system have been rearranged into a three domain system. So these three domains of life are actually bacteria, archaea, and eukaryotes. So based upon the ribosomal small subunit, rRNA, so first of all, the eukaryotes have two type of ribosomes. One is 70S, which are present in organelles, for example, chloroplast and mitochondria. So mostly prokaryotes, because now we know, according to endosymbiotic theory, that the bacteria, proteobacteria and cyanobacteria. So proteobacteria gave rise to the mitochondria, and cyanobacteria gave rise to the chloroplast. So because both came from the bacteria, so mostly bacteria and the prokaryotes are having 70S ribosomes, whereas eukaryotes are having 80S ribosomes. Now, what are ribosomes? Ribosomes are the cellular machinery over which proteins are biosynthesized. So these are cellular machines for translation, so in which the mRNA sequences are converted into proteins. So these ribosomes, whether it is a 70S in case of bacteria or in case of mitochondria or chloroplast, or whether they are 80S, for example, in case of eukaryotes, they are made up of two ribosomal subunits. So let us discuss. So they are made up of two ribosomal subunits. One, in case of 70S ribosome, which are present in mitochondrial chloroplast and prokaryotes, bacteria, it is made up of 30S and 50S. S is nothing but the Swede-Works unit. Okay, so if you add, it will be 80, but actually the 70S is made up of 50 and 30S, whereas the 80S is made up of 60 and 40S. So please do not get confused by this addition, because if you add 60 plus 40, it will be 100. But actually, when these two bind, it will give a subunit, means the complete ribosome, which is having the molecular weight is 80 Swede-Works unit, 80. Okay. So there are two different type of ribosomes, 70S and 80S. 70S is made up of 50S and 30S, whereas in eukaryotes, so 80S is made up of 60S and 40S. Now, these two different subunits, this is called larger subunit, this is called small ribosomal subunit. Okay, so large subunit is made up of in 70S, or either I say 50S ribosomal subunit is made up of 23S rRNA and 5S rRNA along with some proteins. So these are ribonucleoproteins because it is made up of rRNA and proteins. Whereas the 60S subunit is made up of some 40 proteins and three different type of rRNA. So instead of 23 here, it is made up of 28S rRNA, 5.8S and 5S rRNA. So these both are same, but the difference is in these two rRNA, and some additional proteins are present, so it will make an heavy subunit, 60S instead of 50S. Whereas, okay, so when we try to classify the three domains of life that are called eukaryotes, archaea, and eubacteria, so we are interested in these small ribosomal subunits, which are 30S and 40S. So 30S is actually made up of 16S rRNA and 21 associated proteins. We will take only 16S rRNA, and we will take 18S rRNA from this particular small ribosomal subunit. Now, based upon the 16S and 18S ribosomal subunit rRNA sequences, because they are having some signature sequences, these signature sequences are nucleotide sequences based upon which the 5-kingdom classification system have been rearranged to give rise to this new classification, which is called as three domains of life. Okay, so bacteria, that is today it is called eubacteria, archaea, and eukaryotes. Okay, so we will discuss how they have classified the bacteria, archaea, and eukarya. And one more thing before getting into the text, I would like to tell you that this archaea is more close to eukaryotes. If you look at this phylogenetic tree, so here it is having some last common ancestor that is called Leuca. So here. So this is some ancestor. This is actually called Leuca, last universal common ancestor, Leuca. From there, actually, there is a branch here. So from this branch, there are two major groups. One is bacteria. So bacteria have diverged early in, okay, so this is the time in millions of years. And if you see, the bacteria or eubacteria have diverged from this particular branch. Then there is another branch point here. So this major branch then differentiated the eukaryotes and archaea. So archaea went this route, and eukaryotes further evolve and give rise to entamoeba, slime molds, animals, fungi, plants, ciliates, flagellates, trichomonads, diplomonads, and microsporida. So these different eukaryotes have been evolved from this particular line, whereas now you are able to understand that this archaea and eukaryotes are more related to each other if we try to compare them with the bacteria, because bacteria is completely different. They have diverged earlier in the evolutionary period. Therefore, archaea and eukaryotes are more closely related to each other, and this similarity can be seen in their lipid makeup of their plasma membrane, okay, and also in their signature sequences, okay. So now we will discuss how they have classified into these three domains first. Okay, let's start from the beginning either. So the prokaryotes or the bacteria, pro means before, karyon means nucleus, okay. So the consist of eubacteria and archaea. So both eubacteria and archaea, which are the two domains of life, have been coming under the prokaryotes. So both are prokaryotes. See again, if this is not clear. So bacteria and archaea are the two different domains of life, but actually they are coming under the same class that is called prokaryotes, PK, whereas we are having eukaryotes, which are completely different because they are having a membrane bound nucleus. So, and the organelles. So they are having compartmentalization within the cells and also having a defined nucleus. Therefore, they're called eukaryotes, whereas the bacteria and archaea do not have the nucleus or the defined. There are some exceptions also. I will tell you the exceptions also, but mostly prokaryotes do not have the membrane bound nucleus. Therefore, they are coming under this prokaryotic. Also, as we have studied the five kingdom classification system, so both bacteria and archaea have been evolved from the Monera. Okay, whereas all the remaining four kingdoms are making the single domain of life that is called eukaryotes or eukarya. So the Monera have been differentiated into bacteria and archaea, but this Monera is actually for prokaryotes or bacteria. Therefore, both are coming under the prokaryotes. So there is a query. Let me see. What is the query? Priyanshi is asking, can prion replicate? Yes, these prion can replicate. Means not in the way you are thinking, like semi conservative, as you have studied in case of DNA, but they replicate in the sense that they will infect other proteins so that the other protein will change the conformation. They will become alpha, from alpha to beta. So they will be infected, and then again these infected proteins will continue to infect the other proteins. They will convert the conformation of the alpha sheets in alpha helix into the beta pleated sheets, so that they will continuously misfold the proteins. Okay. Trishla is having a question. Please write here in the chat box, Trishla. Okay, please write your question somewhere. Actually, it will disturb my class. Please ask at the end of the class. Okay, So if you want to ask your questions, please write them somewhere so that you can ask. I will give you some time at the end of class, no worries. Okay. So actually we need to study this actually. Okay, so the both eubacteria and archaea, they are coming under prokaryotes, and actually they also signify, or they also tell about another kingdom called Monera. So Monera has been divided into eubacteria and archaea depending upon their signature sequences. And okay, so what are the major differences between the bacteria and archaea? So we have already discussed though. First thing is they differ in their molecular makeup. For example, their lipids are different, their cell wall are different. Also, their signature sequences are different. So therefore, they are completely different. And the archaea, because they are having different lipid and cell wall components, molecular makeup is different, which help these archaea to survive under very harsh environmental conditions. Okay. तो इन्हीं की वजह से वो बहुत ही ज्यादा हार्श इन्वायरमेंट में भी सर्वाइव कर पाते हैं. नाउ वी विल डिस्कस व्हाट आर द मेजर डिफरेंसेस बिटवीन अ बैक्टीरिया and eukaryotic cell, but because there are some differences in eubacteria and archaea also. So first major difference is in the cell wall. So the cell wall in case of bacteria is made up of peptidoglycan, and whether it is gram positive or gram negative bacteria. So in gram positive bacteria, the cell wall is thick, generally 70 to 80 nanometer. Okay, but in gram negative bacteria, because the gram negative bacteria is having two plasma membrane, I told you earlier also, actually the gram negative bacteria is having two plasma membranes. One is called outer membrane, which is present outside the cell wall. So the gram negative bacteria, which is having two plasma membranes, having the peptidoglycan cell wall, but this is thin uh, below the outer membrane. It is generally 2 to 7 nanometer thick. So this is thin in comparison to gram-positive bacteria. So this is the major difference between the gram-positive and gram-negative. Now, if you try to distinguish try to compare the bacteria, eubacteria with the archaebacteria. So in archaebacteria, the cell wall is made up of pseudopeptidoglycan. So let me annotate this so that you can remember. So pseudopeptidoglycan. Now, actually, the major difference is in this. The bacteria is having this peptidoglycan, whereas the archaea is having pseudopeptidoglycan. This peptidoglycan is also, you can found somewhere that it is written like murin. So murin or peptidoglycan is same. And if you put the pseudo term here, this will become pseudomurin. So this type of cell wall, pseudomurin or pseudopeptidoglycan, is present in the archaea. Now you would, I do not know whether you know or do not know this. Actually, I have discussed this earlier also. So this pseudopeptidoglycan is made up of, okay, first of all, you should know the peptidoglycan is made up of two components. One is called NAM, one is called NAC. And they are repeated a number of times. And this link, which is present between the NAM and NAG subunits. So what is NAM? NAM is N-acetyl muraminic acid, whereas this is N-acetyl glucosamine. So they are connected via link. This is called beta-1,4 glycosidic link. So this is called beta. Sorry, I cannot write beta like beta because I'm writing normally. So beta-1,4 glycosidic bond, which is present in this peptidoglycan, which is present in the eubacteria. So first I'm telling about peptidoglycan. Now I will tell you what is the difference between peptidoglycan and pseudopeptidoglycan. So this is glycosidic bond. Now we will come to pseudopeptidoglycan. So this is the normal scenario. In case of eubacteria, this peptidoglycan cell wall is made up of NAM and NAG subunits, which were connected via beta-1,4 glycosidic linkages. Whereas in pseudopeptidoglycan, which is present in archaea, they are made up of NAT, NAT, that is N-acetyl talosamine acid, and NAG. And here, this link which is present between the NAT and NAG is actually beta-1,3 glycosidic bond. Okay, so beta-1,3. So now you can think like, okay, so here it is having beta-1,3 glycosidic bond, here it is having beta-1,4 glycosidic bond. So if you treat the peptidoglycan or the eubacteria with lysozyme, which is present in our tears, in our secretions, okay, so it will lyse the beta-1,4 glycosidic bond. So normally the eubacteria will be lysed. It will undergo lysis, osmolysis after treatment with the lysozyme. Whereas because the archaea is having beta-1,3 glycosidic bond, it is resistant to the lysozyme. It cannot be cleaved with the help of lysozyme, because this is also having some different component, that is not NAM here, this is NAT, N-acetyl talosamine uronic acid, instead of N-acetyl muraminic acid. So there are differences between these archaea and bacteria in terms of their lipids, in terms of their cell wall, and therefore they have been classified in under two different domains of life. One is called archaebacteria, one is called eubacteria. The informal name bacteria is occasionally used too loosely in the literature. Somewhere you will find that we generally call them bacteria, but in the literature it is referring to prokaryotes, and this bacteria, meaning prokaryotes, is comprising both eubacteria and archaea actually. So now you are aware that these are two different domains of life, therefore the care should be taken to interpret its meaning in any particular context. Prokaryotes can be distinguished from the eukaryotes in terms of their cell structure and molecular makeup. So there are many differences which are given in the form of table in this particular table. So membrane-bound nucleus, which is present only in eukaryotic cells but absent from the prokaryotic cells. DNA complex with histones. We know the histone proteins are present in eukaryotes, for example, in animals. But when we compare them with prokaryotic cells, the histone proteins are not present. But to stabilize the DNA, to wrap the DNA in a region that is called nucleoid region, where most of the bacterial cell DNA or genetic material is present, they are complex with some positively charged proteins that is called polyamines, for example. Okay, but the histones, proper histones, which are present in the eukaryotes are not present. They are absent from it. Number of chromosomes. This is one chromosome, circular chromosome. But here, this is generally more than one chromosome. For example, in case of humans, we are having 22 plus 1 XY or XX chromosome. Mitosis and meiosis are absent from the prokaryotes, but we are having both. Sterols and plasma membrane. So normally our cells, different somatic cells, undergo mitosis, whereas our germ cells are undergoing meiosis to give haploid cells. Sterols in the plasma membrane, they are present in archaea, they are present in eukaryotic cells, but they are absent from the prokaryotes, except there are some exceptions, and we are not generally talking in terms of exceptions, we are talking in general way. So generally they are absent from the prokaryotic cells except in mycoplasma. You know, mycoplasma is PPLO, pneumonia like organisms. These are smallest non bacteria because they do not have a cell wall. Ribosome. As we have discussed already, these prokaryotic cells have 70s ribosome, which are made up of 50 and 30s subunits. Here they are made up of both present, generally in the cytosol, 80s ribosomes are present, whereas in organelles, for example, in mitochondria and chloroplast, uh, in case of plant cells, they are having 70s ribosomes as well. So okay, one more thing you need to remember here, though you have discussed. So these are made up of two subunits. One is 50s and one is 30s plus 30s. Sorry, this is too weird to write like this. So 50 plus 30 set. So this is our 70s. Now, one more important thing to remember from here is this 50s is made up of 23 rRNA, 23S, sorry, plus 5S rRNA. Oh, sorry. Whereas this 30S is made up of 16S rRNA. Clear? Whereas only this 30S is made up of 8 and this 16S rRNA and associated proteins with each subunit. Now this 80S is made up of 60 plus 40 S ribosomal subunits. In this case, the 60S subunit is made up of 3 rRNA and associated proteins. So one is 28S, one is 5.8, and one is 5S rRNAs plus associated proteins. Whereas the 40S is made up of 18S rRNA. So this 40S is made up of 18S rRNA, which are okay. So this 18S out of this 40S small ribosomal subunit and the 16S out of this 30S ribosomal subunit is generally used as signature sequences to compare the different animals. Now, both what are the advantages? So there are some, if you compare thousands of organisms, right? So there should be some similarities, there should be some variations. Both similarities and variations are accounted for the classification of organisms. We will discuss this. Now there is unit membrane bound organelles with, for example, mitochondria and chloroplasts, because these are itself derived from prokaryotic cells. So they are absent from prokaryotic cells. They are generally present in symbiotic association because these are semi-autonomous. You know the term semi-autonomous because some of the proteins are synthesized from nuclear genome or DNA, nuclear DNA of the host. Therefore, they are called semi-autonomous, not the autonomous ones. But here they're present in the symbiotic association. Cell wall. Cell wall is found in prokaryotic cells, and in eubacteria it is made up of peptidoglycan, which is made up of NAM and NAG linked via beta-1,4 glycosidic bonds. Here they are made up of cellulose in plants, chitin in fungi, and it is absent from the animal cells. Animal cells do not have cell wall. Hope you are able to understand those major differences between a prokaryotic cell and an eukaryotic cell. If this is clear, now there is phylogenetic overview, how we can classify all the organisms over Earth into three domains of life. So historically, prokaryotes were classified on the basis of their phenotypic characteristics. So initially, when Whitaker gave the 5-kingdom classification system, so Whitaker has classified them in terms of their phenotypic, their characteristics. Okay. Now the prokaryotic taxonomy therefore involved measuring a large number of morphological as well as biochemical characteristics. So उनको classify करने के लिए उसको क्या क्या करना पड़ा? Morphology study करनी पड़ी. You need to observe the morphology, their differences in the structures, and biochemical characteristics, for example, ability to grow over different type of cultures, subcultures, cell wall structure, antibiotic sensitivity, and many others. Okay, so the question is from where these antibiotic sensitivities are coming. So, you know, some of the microorganisms are resistant to some of the antibiotics. It is because most of the bacteria or microorganisms have plasmid, and this plasmid is having these antibiotic resistant genes, for example, ampicillin resistant, tetracycline resistant, chloramphenicol resistant genes. If it is having that particular resistance gene present within this plasmid, that particular microorganism or bacteria will be resistant to that particular antibiotic. It is not having that particular plasmid or that particular gene for the resistance. This is said to be sensitive for that particular antibiotic. If you give the antibiotic, it will kill the microorganism. Cell wall structure, this we had discussed in case of gram-positive, gram-negative bacteria. Ability to grow different substrates. We will discuss this in upcoming texts because there are different types of culture media. For example, Luria bertini, LB, McConkey, and so on. So we will discuss them also. So they are used to culture or subculture different type of organisms depending upon our requirement. Fine. So what else? So this classification, which is based upon morphology and biochemical characteristics of that particular organism, contrasts with the classification of eukaryotic organism, for which phylogenetic evolution-based classification was possible through the availability of the partial evidences. Okay, so now the question is, because we can classify humans or the eukaryotic organisms which have fossil evidences, but we cannot classify the microorganisms depending upon their evolution because we do not have their fossils. So these fossil records or evidences are important whether we want to classify, you know, or give them classification depending upon their evolution. So the major revolution, now what is the solution to this particular problem if they do not have the fossils, if we do not have the fossil evidences? So the major revolution in this classification of prokaryotic organism occurred with the realization that the evolutionary relationship could be deduced on the basis of differences in the gene sequences. If we have differences in the gene sequences or the signature sequences, we are able to classify the microorganisms as well based upon their evolution. So the rRNA okay, so the ribosomal RNA from small ribosomal subunit, as we had discussed, either from the 30S or from the 40S. Okay, small ribosomal subunit. So this 16S ribosomal rRNA from eubacterial and archaeal cells and 18S from the eukaryotes have become the molecules of choice for interfering the prokaryotic phylogenies. So because we do not have any option, therefore, we need to classify these microorganisms depending upon their 16S or 18S, for example, in case of eukaryotes, gene sequences. Small subunit RNA, that is called SHU, small subunit SU4 subunit, rRNA, are an integral part of the ribosome, we know. And the genes encoding these rRNAs change very slowly with time and do not appear to be subject to horizontal gene transfer. Because they are generally present in the ribosome, and ribosomes are an integral component of any cell, and generally there are very few changes. They do not appear to be subject to the horizontal gene transfer in case of this small subunit ribosomal RNA. Therefore, we can classify depending upon this SSU, small subunit ribosomal rRNA, contains certain sequences that are variable among organisms and other regions that are quite similar. Now, so as I told you, both are important. The variations among the organisms in the certain sequences, and also there are some similarities, quite similar in some organisms. Now, what is the benefit of variable regions? Because there are some variable regions among the certain sequences. So, variable region enable the comparison between very closely related organisms. Suppose you are having the closely related organism. Now, depending upon these variable sequences, you can classify them, you can differentiate them. Whereas the stable sequences allow the comparison of distantly related microorganisms. If you are having quite similar sequences, now depending upon these similar sequences, you can classify very distantly related microorganisms or microbes. So comparative analysis of these small subunit rRNA sequences from thousands of organisms once upon a time has demonstrated the presence of oligonucleotide signature sequences. These signature sequences are important from the point of view to classify them into three domains of life. These are short, conserved nucleotide sequences that are present in one taxonomic group but not in the other group. Because these signature sequences merely are responsible for classifying different microorganisms, different organisms into three domains of life, they are present in one taxonomic group, but they are not present in the other groups. Therefore, we are able to classify the organisms. Thus, the signature sequences found in eubacterial rRNA are rarely or never found in the archaeal rRNA. Or this can happen vice versa. Likewise, the 18S rRNA or eukaryotic system, because this is 18S and this is generally present in 40S ribosomal small subunit in eukaryotes, we have the signature sequences that are specific to this particular domain only, domain eukarya. So depending upon these differences in signature sequences, you can classify all the organisms into the three domains of life based upon ribosomal RNA signature sequences. And that was exactly done by this particular scientist, Carl Woese, who proposed this three-domain classification system. Carl Woese proposed a radical reorganization. So it was not new, it is a reorganization of five kingdom classification system into the three domains. In his classification system, Carl Woese placed all the four eukaryotic kingdoms, that is called Protista, fungi, plantae, and animalia, into eukaryotes or eukarya, which is known eukaryotes. And then he split the former kingdom Monera, as I told you already, into the two groups, eubacteria and archaea, depending upon the differences in their signature sequences, 16 srRNA gene sequences. So unlike Whitaker five-kingdom classification system, whose three-domain system organizes biodiversity by evolutionary relationship. If somebody asks you what kind of classification it was, so the reorganization or the kind of biodiversity, it was classified into the three domains of life depending upon the evolutionary relationships. And this phylogenetic tree, which is discussed after the paragraph, is actually showing the evolutionary relationship between different type of organisms. For example, they have common ancestor, then the bacteria have evolved, then the archaea is further differentiated, and eukarya is giving rise to this particular branch. Okay. Now, so this we have discussed already, no problem. You can read this, there is nothing important. So according to this universal phylogenetic tree, archaea and eukarya share a common ancestry. You can see from here that because this branch is after this particular branch, therefore the archaea and eukaryotes are having this common ancestry. They are having this common ancestor at this particular branch. What type of organisms were whatever type of organisms were present over this particular branch, they are common ancestors for both these archaea and eukaryotes. Okay, if you look into the first page of Prescott itself, there is a picture of stromatolites. Okay, so what are the, okay, so here, if you can see this particular picture, you can see the huge rocks, but these are not rocks actually, these are cyanobacteria. Cyanobacteria in the sense they are fossilized stromatolites. What are the stromatolites here? So they are today called stromatolites. They are actually present in the Western Australia. Each stromatolite rock-like structure, which is having around 1 meter diameter, containing a layer of cyanobacteria. So you can see, you can get the fossils of cyanobacteria from here, from these stromatolites, which is a kind of evidence of the existence of photosynthetic organisms early in the history of life over Earth. So today they are the major evidences that earlier the cyanobacteria was a major organism for the photosynthesis over Earth. And the cyanobacteria have contributed a major portion in the oxygenation, you can say, the evolution of oxygen over Earth, or in photosynthesis. Then again, so the next paragraph is actually about the origin of eukaryotic semi-autonomous organelles. As we have discussed the two semi-autonomous organelles, one is mitochondria, one is chloroplast. As I told you, the mitochondria have been evolved. So look here, this is bacteria, and it is having proteobacteria and cyanobacteria. Now you need to remember the proteobacteria, alpha proteobacteria, that is also called Rickettsia prowazekii, actually one specific microorganism in this particular class, proteobacteria, gave rise to mitochondria, whereas cyanobacteria is giving rise to, because the cyanobacteria is able to do photosynthesis, and it will give rise to chloroplast. So mitochondria and cyanobacteria will give rise to chloroplast. Fine. Now we will discuss how these two organelles, mitochondria and chloroplast, have been evolved, and what type of hypothesis or the scenario have been in the scientific community emerged as evidence for these two semi-autonomous organelles. So first of all, this kind of semi-autonomous, you should able to understand the word semi-autonomous. So something which is not autonomous. So this is semi, because they are able to synthesize only some of the proteins of their own over their 70S ribosomes, whereas some of the proteins which are synthesized by cytosolic, cytoplasmic ribosomes, 80S ribosomes, and the mRNA is coming from the nucleus to the transport, extranuclear transport. So it will help in the synthesis of some of the proteins for the mitochondrial chloroplast. They are transported across the two membranes. So also the proof that these are semi-autonomous and these are symbiotic in nature is coming from because they are having two plasma membranes. One is outer membrane, one is inner plasma membrane or inner mitochondrial membrane, for example, as we have discussed. So the electron transport chain is happening in inner mitochondrial membrane, and they are also having an outer mitochondrial membrane. So the gap between outer and inner mitochondrial membrane is the reason where your potential is generated and H positive ions are accumulated. So mitochondria and chloroplast evolved from the free living bacteria, either from the proteobacteria or cyanobacteria within eukaryotic host cells. This is called endosymbiotic hypothesis. Endosymbiotic in the sense because they start living, these bacteria started living inside the eukaryotic cells. Therefore, this is called endosymbiotic association or hypothesis for the generation of these two organelles, mitochondria and chloroplast. For the past several decades, various endosymbiotic models have been proposed, and two of them are popular today. One is called Archijohn scenario. One is called symbiogenesis scenario. So first of all, we will discuss what is Archijohn scenario. Archijohn scenario is based upon the hypothesis called hydrogen hypothesis. I will tell you hydrogen hypothesis also. So in this case, because this is archaeozoon, so archaea related term. So you can understand that the host in this case is an archaea. As we had discussed three domains of life. So suppose if the archaea is the host and eubacteria is the organelle or this mitochondrial chloroplast, if it is going inside the archaeal cell, this type of model is called archaeozoon scenario. So this is not the preferred one. This is not very popular, but the second one, that is called symbiogenesis scenario, is popular these days. I will tell you the reason. Okay. So host that acquired mitochondria was an anaerobic nucleus bearing eukaryotic cell that is termed as archaeozoon. Whereas in contrast, okay, so now you can assume this that, so wait a minute, move. Okay, so suppose there are three cubes. This is having 1 mm, okay, so side. This is having 2 mm and this is 4 mm. You can think of this cube can be fitted inside this particular cube and this cube can be put inside this cube. Okay, so same is the case with the symbiotic association or endosymbiotic association in case of mitochondria and chloroplast, because mitochondria and chloroplast are derived from the proteobacteria or from the cyanobacteria. And you know, the bacterial cells are having generally smaller in size when compared to eukaryotic cells. So bacterial cells are generally around 1 micrometer diameter, 1 micron diameter, whereas the eukaryotic cells are generally 10 micron. So they are even larger, but suppose they are 5 to 10 micron diameter. So you can think of a small cell can be put inside a large cell. So therefore, this bacteria make an symbiotic association with this eukaryotic cells, and this lead to this particular hypothesis that is called endosymbiotic hypothesis. Endosymbiotic hypothesis. So first time Lynn Margulis told about this particular endosymbiotic hypothesis, in which she proposed a model that there were two cells. One is a prokaryotic cell and there was an eukaryotic cell. This prokaryotic cell has been engulfed by this particular eukaryotic cell, because this was large in size, this was small in size. So suppose this was engulfed by this eukaryotic cell, and later on they start living in a symbiotic relationship, which lead to the origin of two important organelles within the eukaryote. One is mitochondria, one is chloroplast. Okay. Now, first model is Archeojohn scenario. In this particular scenario, the host, which is acquiring the mitochondria or this proteobacteria, was an anaerobic nucleus bearing eukaryotic cell, and this eukaryotic cell is termed as archeozoon because this was anaerobic. In contrast, the second hypothesis or scenario is symbiogenesis scenario, where an ensymbiotic event involving the uptake of an alpha proteobacterium, that is actually Rickettsia prowazaki. You should remember this particular name because mitochondria has been evolved from this organism only, Rickettsia prowazaki, by an ancestral cell, by an archaeal cell lead to the generation of the mitochondria, followed the subsequently by the evolution of the nucleus and compartmentalization of the eukaryotic cell. So according to symbiogenesis scenario, actually, it was believed that it was an archaeal cell in which who engulfed this alpha proteobacteria, Rickettsia prowazaki, and this lead to the generation of or the evolution of not only the nucleus and also the compartmentalization within this eukaryotic cell. So they believe that this eukaryotic cell have been evolved from the combination of an bacteria, that is Rickettsia prowazaki, and an archaea, which is the host here. So archaeozoal scenario most closely approximated to the classical endosymbiont hypothesis of the mitochondrial origin, whereas the hydrogen hypothesis is an example of symbiogenesis scenario. Now we will discuss what is hydrogen hypothesis, because first one, Archean scenario, is a kind of some classical model, but the symbiogenesis scenario is based upon this hydrogen hypothesis. According to the hydrogen hypothesis, okay, so let me tell you in more details. So hydrogen hypothesis, okay. Sorry. This is also not. Okay, so here. So hydrogen hypothesis is a model which is proposed by these two scientists, Martin and Muller, in 1998. Okay. Describe a possible way in which the mitochondria arose as an endosymbiont. So what are the possible ways? So in the hydrogen hypothesis, the host which acquired the mitochondria was a hydrogen-dependent archaea. This archaea is the host here, and it requires hydrogen. Whereas the proteobacteria, which is giving rise to mitochondria, is some hydrogen uh, releasing bacteria. So hydrogen-dependent archaea, possibly similar to the modern methogenic archaea, which use the hydrogen and carbon dioxide to produce the methane, for example. And a future mitochondrion that was actually a eubacteria or proteobacteria, Rickettsia prowazaki, was a facultative anaerobe eubacteria that produce hydrogen and carbon dioxide as a byproduct of anaerobic respiration. Now, because this proteobacteria will produce carbon dioxide and hydrogen, which is being used by the archaea, which is also anaerobic, and the combination of these two, this archaea and eubacteria, there is a symbiotic relationship between the two started, and this based on the host hydrogen dependence. This is actually anaerobic syntropy. Okay, so this type of model is based upon this hydrogenosomes, which are present type of membrane enclosed organelles found in some ciliates, trichomoniads, fungi, and animals, which are producing or utilizing the hydrogen. Okay, and the hydrogen hypothesis is also the basis for this symbiogenesis scenario, which is a combination of an archaea, which is the host here, and a Rickettsia prowazaki, which is the parasite, And the generation of mitochondria lead to the generation of mitochondria, followed by the subsequent by the evolution of the nucleus, and the nucleus and compartmentalization within the eukaryotic cell have been taking place. The archeogene scenario most closely approximates the classical. Okay, this we have discussed actually. So this scenario was based upon hydrogen hypothesis, which we also have discussed. However, it would seem that symbiogenesis scenario, that is actually the bacterial endosymbiont in an archaeal host, better accommodates the accumulated data which is relevant to the question of mitochondrial origin than does with an archaeogene scenario. So actually the second one, that is symbiogenesis scenario, is more probable and better suited. Okay, meaning of the endosymbiont and the organelles are different because what is meaning of an organelle? Organelle is some organelle is some compartment which is present inside a cell, whereas the endosymbiont, so the usual way to distinguish an organelle from an endosymbiont is by their reduced genome size. So what will happen as an endosymbiont evolve into an organelle? So suppose some organisms start living inside some other organism, for example, in this case, eubacteria, and eubacteria start living inside an eukaryotic cell or inside an archaea. So what will happen? There will be the most of their genes are transferred to the host cell genome. So what will happen? Their genome size will be reduced in case of parasite. So the most of the genes of the parasite will be transferred into the host cell genome. For example, as occur in the case of eukaryotic cells, most of the genes of mitochondria or the proteobacteria or the cyanobacteria in case of chloroplast have been transferred to the nucleus of the eukaryotic cell. Therefore, it is believed that the nucleus have been evolved after this after the symbiosis event. Okay, so that's all for today. We will discuss and start this structure of bacterial cells. As we know, there are different shapes of bacteria, coccus, vibrio, and one is this vibrio is comma shape. Coccus is round shaped bacteria. One is rod shaped and spiral shaped. Okay, these are four major shapes of bacteria. Whereas some other combinations can also happen. For example, strepto for chain, or in case of some organisms, some microorganisms can have more than one forms. They're called, if they are present only in single shape, they're called monomorphic. Whereas if they're found in more than one form or shape, they're called pleomorphic. Okay. Also, there are some other terms which are used. For example, staphylococcus. Okay, so what is the difference between strepto and staphylo? This has been asked in CSIR actually. So strepto is the chain-like organization. So if the coccus make a chain-like structure, so these are called strepto. So different coccus will make a chain so that this type of structure will emerge. Okay, so cannot make the straight lines, but something like this. Whereas in case of Staphylo is something clustered. So if you see the organization like, as you see the bunch of grapes or the clusters, then this type of organization of these microorganisms is called as Staphylococcus. So in this case, what will happen? They will make the bunch-like structures. So if you find something like this, then this is called Staphylococcus. So Streptococcus, Staphylococcus, and we will also discuss, for example, depending upon number of, so Diplococcus, only two, Diplobacillus, sorry, this is Diplobacillus. So we need to make the rods. So there are two rods which are linked together. They're called Diplobacilli or Diplococcus if they are two bacillus. And so on. Okay, so we will discuss these also as it is given here. Bacillus is rod shaped, coccus is spherical, Vibrio is comma, and Spirilla is some having helical shapes. So different microorganisms are having different shapes. So under microscope, they can be distinguished from each other. Also, they can be stained to increase their contrast to understand their morphology better. So therefore, they can be stained with the different dyes, as in case of gram stain, or some acid fast stain, for example, in case of Mycobacterium tuberculosis and lepri, because they cannot be gram stained. So we will discuss these staining procedures and acid fast staining, which use the Gill-Nelson stain in case of mycobacteria. Okay. Now, if you have any query, this is the time to ask.